Okay, welcome to our first video on gluconeogenesis. We will have a two-part series here. And so here's our overall big map, and then we're going to zoom in on the section we've been looking at. And so our last big topic was glycolysis, where we took glucose and converted it to pyruvate. And so gluconeogenesis is going to be essentially the backwards version of that. Now, when you think about glycolysis, starting with glucose, there are numerous sources of glucose, right, that we could have started with. So starch, lactose, glycogen, there's all these other sources that can feed in for glucose. But when you are in a fasting or starvation state, those sources are no longer available. They'd be all depleted, and your brain depends on glucose. So our bodies need a way to synthesize it when we need it, and that's what we're going to be looking at. So glucose can be synthesized from non-carbohydrate precursors. Pyruvate is the one we're really going to focus on. Also carbon skeletons of some amino acids and glycerol. So looking kind of briefly at these. So pyruvate we know was a product that then got further broken down or converted after glycolysis. And one of those things that happened was we could form lactate. Generally in our muscles. So over here, we're looking right here. And remember that anaerobic process that made lactate was important for regenerating the NAD plus for the glycolysis. And what can happen now is lactate can enter that bloodstream, and if energy is needed in other parts of the body, lactate can be converted to pyruvate and then move into other energy metabolisms. Or if the energy needs of the body are met, lactate can be taken up by the liver, right? So over here we're in the liver, converted back to pyruvate, and then shuttled into this gluconeogenesis pathway that we're going to look at for forming glucose. Now the other two we're not going to look at in depth right now. Carbon skeletons of some amino acids. We'll talk about amino acids more later when we talk about amino acid metabolism. Okay, but those could be converted into gluconeogenic intermediates. And glycerol. So think about when we looked at glycerol, we talked about it with the triacylglycerols, the lipids, right? And those can be converted back, and we'll look at just a sec where that would come into that glycolysis or gluconeogenesis pathway. But essentially, we can take glycerol, add that phosphate, so this shouldn't be a surprise, right, when you see glycerol kinase, so we're adding a phosphate. And then this dehydrogenase over here, notice that we drop down to this we oxidize it, right? We get this carbonyl. That's the word I'm looking for. So if you remember, we talked about when you see dehydrogenase, you can really think redox reaction, and we can see that oxidation process. And we're going to look at where that falls in. You don't need to memorize these specific steps, but it's good so you can see how these different biomolecules can be interconverted into different metabolic pathways, depending on your body's needs. All right, we are going to focus in now. Gluconeogenesis. Synthesis of glucose. So we're now synthesizing glucose from pyruvate. The major source of this is in the liver. Although it can occur in the kidney, we're really going to focus in on the liver. All cells can undergo glycolysis, but only the liver and the kidney can synthesize the glucose. And gluconeogenesis is especially important during fasting or starvation, right? So your brain needs glucose as its primary fuel, and it's also extremely important for red blood cells. So let's look. So what we have here on the left is our glycolysis. That should look familiar. And on the right is gluconeogenesis. So here, let's label that. So we've got glycolysis. And over here we have gluconeogenesis. And we've got them side by side. We're going to go through these steps in detail, but let's just make some kind of big comparisons here. First of all, notice our starting and ending products are the same. They're reversed. So in glycolysis, we're starting with glucose. So notice that is, where is the pointer I want? There we go. That's right here in the circled in red, and we see the same thing over here. That's our ending point for gluconeogenesis. And then in glycolysis, we're ending with pyruvate, which is now our starting point. Okay, I told you we'd look at where glycerol came in. So here's glycerol, right? So it was the backbone to those triglycerides, and that glycerol can enter in right here to make that dihydroxyacetone phosphate that is converted to that glyceraldehyde 3-phosphate. So it drops in here, and then depending on your body's needs, right? Either it's going to continue on down to make glucose if we're in the gluconeogenesis pathway, or if we're in the glycolysis pathway, it would head towards pyruvate. If you remember, there were 10 steps in glycolysis, and only 3 of them were regulatory or irreversible steps. Most of them were reversible. And so for those 7 reversible steps, we're going to see the same step with the same enzyme that is working in our gluconeogenesis pathway. And I've highlighted those with different colored arrows. So for example, in glucose, the first step here we'll come back and talk about because that was one of our regulatory steps. But the second step, this isomerase enzyme, phosphoglucose isomerase, we're going to see that exact same enzyme down here. But now it's going to go backwards. It's going to convert the glucose 6-phosphate to the fructose 6-phosphate. All right, our third step we'll have to come back to in glycolysis because that was irreversible. But here, this aldose and this triose phosphate isomerase, you'll see those exact same ones working for us over here. And it goes on through, right? Glyceraldehyde 3, phosphate dehydrogenase. Look, there it is. And so you can see that those steps that were reversible in glycolysis, well, they're just going to run in reverse in gluconeogenesis. All the substrates are the same, so fructose 6-phosphate, fructose 1,6-bisphosphate. The only big difference we're going to see there is that right here, to get from the phosphoenol pyruvate to pyruvate was one step, one regulatory step in glycolysis, and it's 2 steps in gluconeogenesis. So gluconeogenesis ends up being 11 steps total. Let's look at those regulatory enzymes. So every place we had a regulatory enzyme in glycolysis, we're going to expect to see one in gluconeogenesis. So for example, right down here, where we went from phosphoenol pyruvate to pyruvate, we're going to need different enzymes to get us back from the pyruvate to the phosphoenol pyruvate. Okay, because this one right here, so this was not reversible. So we're going to need new enzymes to carry that one out. Okay, the one, our PFK enzyme in green, we're going to need a new enzyme. If you remember, this one was a kinase. We were adding a phosphate. Well, in this case over here, we're taking away a phosphate. So we'll have a phosphatase. So it's just the reverse. It's taking away a phosphate. Still named for the reactant, fructose 1,6-bisphosphatase. Now we're adding that ending. And then we had hexokinase as our first step in glycolysis. And down here, we'll have the glucose 6-phosphatase. So again, we were adding a phosphate there. We're taking one away here. All right, we're going to look at some of those in depth. So let's start with the conversion of pyruvate into phosphoenol pyruvate. So it requires two enzymes because it's two steps. The first step is going to occur in the mitochondria. Okay, and it's going to catalyze this oxaloacetate. Okay, so this is the only substrate that we didn't really see at all in glycolysis, but we will see in gluconeogenesis, right? So right here. So we're taking our pyruvate, going to that oxaloacetate. And then in our second step, which occurs in the cytoplasm, so we're going to have to get this stuff out of the mitochondria into the cytoplasm. Now we're going to go to our phosphoenol pyruvate. So we're going to focus in on these two steps. So when we look at what is happening here, we've got our 3-carbon, 1, 2, 3 pyruvate, and over here we have 1, 2, 3, 4 carbons. So we're adding this carboxylate group, this COO minus. Okay, this is an example of coupling two processes. So notice we had to hydrolyze ATP to provide the energy to essentially force that carboxylate onto this compound. So then the question becomes, how is that happening? Like, is it just ATP is breaking apart nearby and that energy somehow floats over? So we want to look at that in depth because that's an important question, is how in the world does that happen? And it turns out it's not a simple process. It actually occurs in 3 steps. It has a cofactor, so the enzyme, pyruvate carboxylase, has biotin as a cofactor, also needed so that the enzyme can work. And it occurs in these 3 stages. So first, the bicarbonate in solution reacts with ATP. And what happens is we see this phosphate group bond to the bicarbonate. So now we've got our ADP. So we took that phosphate off of here, but we stuck it onto that bicarbonate. In the second step, we have our enzyme, which has its cofactor bound to it, that biotin. Okay, and it forms a covalent bond to the carboxylate group, okay, to that COO minus group, and frees the phosphate. So when you look over here, now we've got the enzyme with the biotin, and it's taken that COO minus group, and we've released some phosphate. And then in the third step, we had this enzyme biotin carboxylate piece, and it reacts with pyruvate in an active site of the enzyme, okay, to form that oxaloacetate, which looks like it is misspelled right here. Yes, we missed an A, but you've got it spelled up there. So when we look at the 3 steps, when you look at the overall reaction up here, it kind of looks like, wow, ATP is just going to ADP totally separate. But when we look at the 3 steps, we can see how those are chemically linked, right? That the energy from this high phosphoryl transfer potential ATP is essentially now trapped in this new bond with this phosphate, okay, to the bicarbonate. And then that gets passed along to this bond here that then finally gets passed along as this reacts to form the oxaloacetate. So let's keep looking at this. When we think about this, this enzyme, pyruvate carboxylase, it is a homotetramer. So it has 4 subunits that are all identical. This entire drawing right here is one subunit. That can be confusing because it looks like 1, 2, 3, 4, and we said it was a tetramer. But this is one single subunit, and there would be 4 of these, all identical, as a part of our pyruvate carboxylase. And when we look, each of the subunits has 4 domains. Okay, so they're shown in different colors. So the blue, the pink, the gray, and the yellow. So let's think about how this works in conjunction with those 3 steps we just looked at. The blue area over here, we're going to call that our first domain because that is essentially where this first reaction is going to happen. So this biotin carboxylase piece, the blue piece over here on the left, okay, will have an active site where the bicarbonate becomes essentially charged with that phosphate. So it will have an active site that takes our bicarbonate and our ATP and helps them link that phosphate onto the bicarbonate. So that's going to occur somewhere over here in this blue one. Then this pink one, clearly not really pink in your body, but color-coded pink here is going to be our second step. Okay, this is where the biotin actual piece is, that cofactor piece is in here. And this biotin is essentially, we're going to call it a swinging arm. It's going to swing down here and it's going to pick up this bicarbonate that we just made. All right, and so now we've got this biotin piece attached, and it's going to essentially bring it over, or swing around over to this yellow domain somewhere. All right, and over here now, we're going to see that third reaction happen. So this is where we're going to see that pyruvate, okay, join in to form that with that carboxylate to form the oxaloacetate. So essentially we have one reaction occurring over here. We have this biotin swinging arm that comes down, grabs those pieces and kind of carries them over, right? So we had our biotin enzyme grabbing that carboxylate group and bringing it over here to another active site where it's going to combine with the pyruvate. This gray piece is the tetramerization domain. It essentially holds the 4 domains together. Okay. This is a great enzyme to study because we see this repeat, this use of this cofactor biotin to kind of carry a carboxylate group from one place to another. We see that repeat in a lot of metabolic pathways, a lot of enzymes. So hence why we are taking a minute to look at it here. All right, so the first step, let's go all the way back up here. So our first step was the formation of that oxaloacetate. That's what we just looked at. And we said that that occurs in the mitochondria. So if you think about your mitochondria, right, we have our mitochondria and we have this oxaloacetate being formed. But we have a problem. And that problem is that that oxaloacetate is not easily transported across the membrane out into the cytoplasm, which is where that second step occurs. And so what happens is within the mitochondria, we actually reduce that oxaloacetate to malate. And then that does have a carrier that will carry it out. And then we reoxidize it to get our oxaloacetate back. So it's kind of interesting that we make it, we have to reduce it. We essentially are changing the carbonyl to an OH group. So it now has less bonds to oxygen, so it's reduced. We're carrying it out, and then we're reoxidizing it to get our molecule back. Once we do that, the phosphoenol pyruvate carboxykinase. Lots of words, right, y'all? Now we can see that. We think kinase. We're like, ooh, it's adding a phosphate. So when we look at this reaction now, we're transferring a phosphate from GTP to that oxaloacetate that was made in the first step. And what we'll see is we'll also lose this carboxylate group. So when we look at this phosphoenol pyruvate has a higher phosphoryl transfer potential. So we need the energy from this GTP and energy, it turns out, from this decarboxylation portion. It takes both of those to essentially let us create this new molecule that has a very high potential energy, so a high phosphoryl transfer potential. This bottom piece right here is the net reaction for the first two steps. There is a big energy investment. We used ATP and GTP to get through steps 1 and 2. So there's a high energy investment to get us from that pyruvate to the phosphoenol pyruvate. All right, that's what we just said. Not sure why I have a second slide. Okay, then let's think of what happens after steps 1 and 2. So we've made the phosphoenol pyruvate, and now all of those enzymes from glycolysis that were reversible will work in the reverse direction until we get to the next irreversible step. So let's go back to our chart. So we just did the piece in pink right here, but then all of these other steps, right, were reversible. And so they're really driven by Le Chatelier's principle, the fact that if you are making more of the phosphoenol pyruvate, it'll drive it towards the 2-phosphoglycerate, right? And if we start making more of that, it'll drive it towards the 3-phosphoglycerate, and so on and so forth, until we get down to the fructose 1,6-bisphosphate. And then the enzyme we used in glycolysis, PFK, was not reversible, and so now we need a new enzyme and a new step to get us to the fructose 6-phosphate. Okay, so this is an allosteric enzyme, so remember what that means. It's a regulatory control enzyme. We're going to look at this one in depth in the next video, but essentially, right, something's going to bond to this allosteric enzyme, and we are going to see that decarboxylation, right, or not decarboxylation, we're going to see that phosphate removed, and we can see that in the name, the phosphatase. We're going to see that phosphate removed. The next step was reversible, but then we get to step 11, which was the first step in glycolysis, that hexokinase, but now we need to go backwards, so we need a phosphatase. We got to take a phosphate off. Okay, this occurs only in the liver. Liver is responsible for regulating blood glucose levels, so we need a way to generate glucose. So this is an important control point. Let's look at what this looks like. So glucose 6-phosphate. So notice here, first of all, this is not a cell membrane. This is separating, this is the membrane separating the endoplasmic reticulum from the rest of the cytoplasm in the cell. And remember that glucose 6-phosphate is in our cell, and there's a special transporter that takes it into this endoplasmic reticulum. In there, that glucose 6-phosphatase is a integral protein on the inner surface of this endoplasmic reticulum, and it catalyzes the removal of that phosphate. And then there are separate transporters that will take the phosphate out back into the cytoplasm and the glucose back out into the cytoplasm. Okay, our net reaction is here. You see we started with 2-pyruvate, we get a glucose, All right, and you can see the energy requirements, right? We see quite a bit of that. It takes a lot of energy for your body to take this back to glucose. All right, real quick, just a couple check for understandings. You may want to pause and think about this for a minute. What barrier prevents glycolysis from simply running in reverse to synthesize glucose? And how is that barrier overcome by gluconeogenesis? Those are your questions. So if you've paused and thought about it, let's see how you did. The barrier for this simply running in reverse is those 3 irreversible steps, right, in glycolysis. Irreversible steps of glycolysis. So it was steps 1, 3, and 10, right? And those steps have such large negative delta G values that they really will only run in that direction. That a buildup of that substrate won't allow them to go in reverse. There's not going to be enough substrate to drive it in reverse. The enzymes are unidirectional, so those steps will not run in reverse. We overcome that barrier with the 4 new enzymes of gluconeogenesis. So glycolysis enzymes, glycolysis will run when your cellular needs are taking that glucose and forming pyruvate, and then your cells will turn on and off the appropriate enzymes. If it needs to run in reverse, it'll turn off those glycolysis regulatory enzymes and turn on the gluconeogenesis ones. All right, we will pick this up in the next video to learn more about gluconeogenesis.